So welcome everyone to today's webinar, Metagenomics Part 1, an introduction to library preparation and sequencing. We will discuss the comprehensive workflow for a metagenomics sequencing project. This will include what metagenomics sequencing is, resources for getting started, library preparation methods, sequencing, and a brief overview of data analysis. We will discuss many topics here, so I will include some helpful resources along the way for more in-depth information. What is metagenomic sequencing? Metagenomic sequencing allows us to determine the microbial diversity of a sample, so how diverse the sample is, and identify the taxa present. We may be looking at bacteria, fungi, archaea, viruses, or combinations of these in our samples. With metagenomic sequencing, we will start with purified DNA from a sample source of interest. Samples can be from soil, skin swabs, water, plants, and many other sources. Metagenomic samples may also be referred to as mixed samples, as they contain a mixture of different species. It is very important to use an extraction and purification method that yields highly pure DNA. Metagenomic samples are often prone to contaminants, which can inhibit downstream steps. If possible, it is best to use a DNA purification kit specifically developed for the sample type. As an example, there are purification kits specifically for soil samples. If a specific kit is not available, using kits or methods that have been demonstrated to work with this sample type, perhaps by the vendor or in the literature, is helpful. I will include resources a little later that discuss extraction and purification methods for some of the possible sample types. And then the bulletin at the link here includes information about possible inhibitors and contaminants, their sources, how to check for them, and how to remove them from our samples. DNA samples must be accurately quantified. We recommend using a fluorometric quantification method specific to double-stranded DNA, like qubit or picogreen. You may also want to consider including a control sample, especially when starting out with a new sample type, a new library preparation workflow, or a new analysis pipeline. Metagenomic control samples include commercial mock community samples, or they can be well-characterized samples from your lab or from sample banks. Another sequencing method that can be used with mixed samples is metatranscriptomics, or microbial transcriptomics. This is sequencing of the RNA as opposed to the DNA from mixed samples. We will not discuss metatranscriptomics here today, though if you are interested in this, the link here includes helpful information for starting a metatranscriptomics project. Now, let's discuss resources for getting started with a metagenomics project. We have several curated pages with helpful information for metagenomics projects, including an overview of environmental metagenomics, an introduction to shotgun metagenomics, and a review of publications relevant to microbes and metagenomics in human health. These pages include publications, protocols, and considerations for metagenomic sequencing, and we will discuss some of this information here today. So now that we have our DNA isolated from our samples, purified and quantified, let's proceed with library preparation. Library preparation of our DNA samples enables us to sequence the samples with an Illumina sequencing platform. For metagenomic sequencing, there are two main approaches to library preparation. The first method is a targeted library preparation approach. With a targeted method, we select a single target in the genome, and we amplify and sequence this target. The target region should be an informative region with SNPs and other sequence variants that will help us to identify the species present in the sample. This approach is sometimes also called metabarcoding. The second method is shotgun metagenomics. With this method, we create sequencing libraries from all of the DNA present in a sample. We will discuss both of these library preparation options in the coming slides. We will start with discussing targeted library preparation. Here today, we'll review two methods for this: the Illumina 16S Metagenomics demonstrated protocol for bacterial metagenomic sequencing, and the Illumina fungal metagenomics demonstrated protocol, which is also known as ITS sequencing. We will also discuss how to customize these workflows for different metagenomics target regions. First, let's chat about 16S sequencing. 16S is a bacterial rRNA gene, and it is a popular target for bacterial metagenomic sequencing. The 16S gene has 9 variable regions, and these vary between taxa. These variable regions, or subsets of regions, can be used for taxonomic classification. The Illumina 16S protocol targets the V3 and V4 variable regions. In the Illumina 16S protocol, we use 2 PCR steps to create sequencing libraries. We use 12.5 nanograms of DNA input, and the protocol can be completed in 1 day. We use the Nextera XT V2 index primers during this workflow, which allow us to uniquely index up to 384 samples for sequencing in a single run. In the protocol, which I'll link to a little bit later, we include the recommended primer sequences for targeting the V3 and V4 variable regions of the 16S rRNA gene in a single amplicon. The primer design for the first PCR step includes locus-specific sequences, as well as overhang sequences, which are utilized in the second PCR. This primer pair can be ordered from the oligosynthesis vendor of your choice. Now, let's review the full library preparation workflow. We start with the first PCR, where we amplify the V3 and V4 regions with the locus-specific primer pair from our input DNA. We clean the products from the first PCR, and then we perform the second PCR. In this step, the Nextera XT V2 index primers bind to the overhang sequences that we introduced in order to uniquely index our samples and complete the adapter sequences. We clean the final PCR products, and now we're ready to validate and sequence our libraries. Final 16S libraries, when run on a bioanalyzer, fragment analyzer, tape station, or other comparable trace instruments, will show a fairly large final library product, typically around 600 bases to 630 bases. Depending on the species present in your samples, you may see differences in the size, or possibly double or even triple peaks as opposed to a single peak as shown in this trace. So what if you'd like to target different variable regions in the 16S gene or a different genomic region altogether for your libraries? For customizing the amplified target, we can change the locus-specific sequences in the first primer pair to target novel regions of interest. We can use the primer layout here, which includes the same overhang sequences used in the standard protocol, and replace the locus-specific regions with the new sequences we want to use. The 16S protocol provides these primer sequences along with guidance and considerations for the locus-specific sequence design. The link here will take you to the 16S landing page, which includes the full library preparation guide with the protocol, along with a frequently asked questions document and example 16S data. Now let's take a look at the Illumina fungal metagenomic sequencing protocol. The Illumina fungal metagenomics workflow is a protocol for ITS sequencing. ITS refers to the internal transcribed spacer regions between our RNA genes. There are 2 ITS regions, ITS1 and ITS2, either of which can be used for identification of fungal species. The Illumina fungal metagenomics protocol, as written, targets ITS1. This fungal metagenomics workflow is very similar to the Illumina 16S demonstrated protocol. We use the same two-step PCR workflow, same input amount, and the protocol takes the same amount of time. The primary difference is in the first round of PCR when we're amplifying up the target locus. Instead of using a single primer pair, we use a combination of 8 forward primers and 7 reverse primers. Using a mixture of forward and reverse primers enables us to better amplify from a diverse range of fungal species. We use the same overhang sequences used in the 16S protocol, and these are here. And then we use 8 forward and 7 reverse locus-specific primers upended to the standard overhangs. We want to pre-combine these primers so we have a forward primer mixture and a reverse primer mixture prior to library preparation. Reviewing the full library preparation workflow, we amplify the ITS1 region with our locus-specific forward and reverse primer mixtures. We clean up the first PCR products. We perform the second indexing PCR with Nextera indexing primers. Then we clean up the second PCR products and we proceed with library validation and sequencing. When viewing our final library traces on the Fragment Analyzer comparable trace instrument, we can see a range of variably sized products. And these can be distinct peaks or they can show a broad distribution or kind of a smear. Libraries will typically have fragments between about 225 bases and 775 bases, and the distributions and sizing will vary based on the species present in the samples. The first link here is for the workflow guide, which includes the full protocol and supporting information. The second link is for the data sheet, which gives an overview of the library preparation, sequencing, data analysis, and results. We've reviewed targeted library preparation, so now we'll go ahead and take a look at shotgun metagenomics library preparation. We have 3 workflows for shotgun metagenomics library preparation. The Illumina DNA Prep with Tagmentation Kit, formerly known as Nextera DNA Flex, the Nextera XT DNA library preparation workflow, and the Illumina DNA PCR Free Prep Tagmentation Kit. We will also discuss a 4th workflow, which is not for canonical shotgun metagenomic sequencing library preparation, though it may be of interest for some labs. This one is the Illumina RNA Prep with Enrichment library preparation workflow with the respiratory virus oligo enrichment panel. We'll start with Illumina DNA Prep. So with this workflow, just a reminder, it was formerly named Nextera DNA Flex. So we can use between 1 nanogram and 500 nanograms of input DNA. The full library preparation will take about 3 to 4 hours total. The workflow is automation compatible, and it uses a 2-sided size selection at the end of the protocol for consistent and reproducible library sizing. Taking a closer look at the library preparation steps, we first start by mixing the bead-linked transposomes with the metagenomic DNA samples. The transposome enzymes fragment the input DNA and they add tagging sequences in a process called tagmentation. After the tagmentation, we perform a PCR with Nextera index primers, which uniquely index the samples, complete the adapter sequences needed for sequencing, and amplify the final libraries. Following the PCR, we use a 2-sided or double-sided bead-based size selection to remove the unwanted large and small library fragments and to purify the final libraries. Once the size selection is complete, we can proceed with library validation, quantification, and sequencing. Illumina DNA Prep with Tagmentation libraries have average sizes of approximately 600 bases. Distributions will start around 300 bases and extend to 1 to 2 kb. Here we have the Illumina library preparation support page and the library preparation reference guide, including the full protocol. The Illumina DNA prep can also be used for other applications, and we have documentation for projects from whole human genome sequencing to bacterial isolate sequencing on the product page at the last link here. Additionally, the documentation page includes links for DNA isolation from dried blood spots as well as bacterial colonies. We have application notes for metagenomic sequencing from crude lysates as well as from soil samples. And we have full protocols for DNA isolation from blood and saliva samples using our Flex Lysis reagents in the reference guide on the previous page. The second option that we have for shotgun metagenomics library preparation is the Nextera XT DNA library preparation kit. We use 1 nanogram of input DNA, and this workflow can be completed in about 5.5 hours. Nextera XT uses a one-sided size selection, which removes unwanted small fragments prior to sequencing. In the Nextera XT DNA protocol, we mixed free transposomes with our purified metagenomic DNA. The transposomes fragment and tag the DNA in the tagmentation process. Then we perform PCR with the Nextera XT index primers to uniquely index the samples, complete the adapter sequences, and amplify the libraries. We perform the one-sided size selection to remove unwanted small fragments at any residual free indexing primers. And after that, we're ready to validate and quantify the libraries, then proceed with sequencing. When we review the final libraries, we will see broad library distributions with variable average sizes. Library distributions will start around 200 to 300 base pairs and can extend to multiple kb. We can see different shapes in the distributions, and this is perfectly fine and expected for Nextera XT libraries. Here, we have the Nextera XT DNA support page and the library preparation reference guide, including the protocol and additional helpful information for preparing your samples. To quickly compare these two workflows, both the Nextera XT and Illumina DNA prep with tagmentation workflows are compatible with metagenomic DNA, amplicons, plasmids, and small genome input. The Illumina DNA prep with tagmentation workflow is also compatible with large genome input like human and plant genomic samples. We can uniquely index and pool or multiplex up to 384 samples with both workflows. The input amount for Nextera XT is 1 nanogram of pure DNA, and the input amount for the Illumina DNA prep with tagmentation workflow is between 1 nanogram and 500 nanograms of pure DNA. Next, let's take a look at the Illumina DNA PCR-free prep with tagmentation. For this, we use between 25 nanograms and 2 micrograms of purified DNA. The library preparation can be completed in as little as an hour and a half, though the preparation time does depend on the input material. This kit is automation friendly, and using the PCR-free approach can be helpful for improving coverage in high GC or high AT regions and genomes. With this, we start by mixing our input DNA again with PCR-free bead-linked transposomes, which tagment the DNA. We then ligate single-stranded adapters, which complete the adapter sequences and uniquely index the samples. We purify the ligated products, and our libraries are ready for quantification and sequencing. Illumina DNA PCR-free libraries cannot be visualized with trace instruments like the bioanalyzer and fragment analyzer. We do provide size information in the reference guide from our molarity calculations. Final libraries are quantified with single-stranded qubit reagents or qPCR. Something a little special about these libraries is that they do require a custom read1 sequencing primer, and they may require a custom index 2 sequencing primer depending on the sequencer that you're using. And we do provide these sequencing primers. If you are using a NovaSeq for sequencing with version 1.5 consumables, you do not need to use any custom sequencing primers as they are already included in the sequencing reagent kit. Here, I've linked to the support page, the reference guide, and a bulletin which gives more information on when and how to use custom sequencing primers for the Illumina DNA PCR-free libraries. We do have an upcoming bulletin which discusses the different library preparation workflow options that you can choose depending on your input. And then we have a fantastic dedicated webinar which is available at the final link here, and you can watch it at any time. The last library preparation workflow we will review is the Illumina RNA prep with enrichment tagmentation. In this workflow, we start with RNA extracted from our samples. Then we create cDNA, and we use B-link transposomes to tagment the cDNA. We use PCR to complete the adapters, uniquely index the libraries, and amplify the libraries. We then use biotinylated probes designed against the sequences we are interested in. These probes bind to the library fragments we want to sequence, and we pull them out with streptavidin magnetic beads. We perform an additional PCR step to amplify our now targeted libraries, and then we're ready for quantification, QC, and sequencing. We offer the respiratory virus oligo panel, which targets SARS-CoV-2, multiple influenza strains, and other respiratory viruses. This targeted panel has been validated on the MiSeq, MiniSeq, and NextSeq sequencers. The support page and reference guide are linked here, along with an application note that reviews characterization of respiratory viruses with this panel, and it includes an overview of data analysis results as well. So now that we have prepared our libraries, we can go ahead and proceed with sequencing. Our first question is where to sequence. So you may have the sequencer you'd like to use in your lab already. You may instead want to use a sequencing service or core facility. These facilities may be academic or non-academic providers. And each facility will have information on how to submit samples for sequencing, and many offer library preparation services as well. If you are looking for a sequencing service provider, we do have a list of providers that have been certified by Illumina. The link here gives more information on Illumina Propel certified providers. So next, we need to choose the right sequencer to use. Different sequencers give different amounts of data. We will need to know how much data we need per sample and how many samples we will sequence to determine the best sequencer for the project. If you are submitting your samples for sequencing, core facilities will likely give sequencer recommendations as well. So before we sequence, we need to know how much data we want for our samples. Data needed may also be referred to as coverage or reads per sample. The amount of data needed will depend on the library preparation method used, the source of the samples, the expected complexity of the samples, and the analysis workflow that will be used. Published literature is a great resource for data and coverage recommendations, and technical support may be able to assist with data and sequencing recommendations as well. When we are ready to sequence, the workflow for setting up the sequencing run will depend on the specific platform that you're using. We have resources on our website to help you every step of the way with this. If you are submitting your samples to a sequencing service provider, the provider will perform the run setup for you after they've received your samples. After our sequencing run is complete, we want to review the run performance and ensure that it performed well. Shotgun metagenomics runs and amplicon-based runs will look and perform differently. For reviewing run performance, we can use Base Sequence Hub online, or we can use the Sequencing Analysis Viewer software, also called SAV, offline. I'll include additional resources a little bit later about Sequencing Analysis Viewer, though in the following slides, we will review the run performance in Base. Overall, they're both pretty similar, but just so you know. So when reviewing a sequencing run, we want to focus on several metrics. We will review 4 key metrics in the following slides. The first metric is the percent passing filter. This refers to the number of clusters on the flow cell or nanowells in pattern flow cells that are giving good signal and will generate data. Second, we will look at the aligned percentage. The aligned percentage refers to the amount of the Phi-X control library present in the sequencing run. The third is the Q-score. Q-scores are how we measure the quality of the sequencing data. A Q-score of 30 or higher is quality data. Fourth, we want to evaluate the percent base. This refers to the abundance of each base, A, T, C, and G, in each cycle of the sequencing run. This will be different depending on if the libraries are amplicon-based, like 16S or ITS, or if they're shotgun metagenomics libraries. For these metrics, there will be different expectations depending on the instrument used for sequencing. If you have any questions about the specific sequencer you are using, please reach out to technical support. So first, let's review sequencing of Illumina DNA prep with tagmentation libraries. For these libraries, we recommend a 2 by 151 base pair read length, so paired insert reads, 151 bases for each. Tagmentation-based libraries have high base diversity, so the inserts in the libraries are widely varied in sequence. We recommend spiking 1% of the Phi-X control library into the library pool before sequencing as a sequencing control. In the following slides, we will evaluate a run with samples prepared with the Illumina DNA prep with tagmentation workflow, run on the iSeq 100 sequencer. Nextera XT libraries will perform similarly. To begin reviewing the run, we will start on the Metrics tab in Base. We can switch our view or tab near the top of the screen. The first metric we'll check is the percent passing filter. For this run, we see that the percent passing filter is 79.95, which is very good for Illumina DNA prep with tagmentation libraries run on the iSeq. Second, we will check the aligned percentage. So the percent of the reads going to the Phi-X control library that we spiked into the library pool before sequencing. Here it is 2.57%. With the Illumina DNA prep libraries, we are targeting about 1%, so this is in line with our expectations. Now we'll go ahead and switch over to the Charts tab, selecting at the top of the screen. We can take a look at the quality of the run here in the Q-score distribution chart. We can select the expand icon in the top right to expand the display. And so now we can see it a bit better. Here we see the percent of the data from the run at or above our Q-score threshold of Q30 is 92.4%, which is fantastic. So now we know our run generated high quality data and it is ready for analysis. If we go back to our Charts tab, the last metric we want to review is the percent base. To view the percent base, we can use the data by cycle chart indicated here. And in the chart drop-down menu, we can select the percent base option. Then we'll go ahead and expand the chart to see it better. And then we'll zoom in a bit more. And here we see the percent base for the run. At every cycle, the instrument reports the percentage of A's, T's, C's, and G's. To help with our review, I've labeled the insert and index reads. These will often look quite different from each other. So for tagmentation-based libraries, we see a slightly spiky pattern at the beginning of both of the insert reads. Following this spiky region, we can see the percent of each base evens out, and we have approximately 25% of each base present at every cycle. This indicates a very well-balanced or high base diversity library pool, which is common of tagmentation-based libraries. The exact percentage of each base will depend on the GC or AT content of the genomes in our samples. So when the percent base plot shows fairly straight lines as the cycles progress, it indicates that the bases are balanced. The index reads are quite spiky in this run, and this can be normal depending on the index sequences we've chosen for our samples. We have millions of different inserts in our libraries, so the insert reads are diverse and balanced. When we get to the index reads, there would now only perhaps be a handful of different sequences. So the overall diversity of the sequences present is dramatically reduced, and this is what causes the spiky pattern in the percent base plot. So we've now completed our review of the Illumina DNA prep with tagmentation libraries run on the iSeq, and we found that the run performed very well. So now we'll review sequencing for targeted amplicon-based libraries. For 16S libraries, we recommend a 2 by 301 base pair read length or paired 301 base pair reads, and that's if we're using the iSeq. If we are sequencing using a different platform, we use 2 by 151 base pair read lengths, so paired 151 base pair reads. With 16S libraries, when sequencing with the paired 151 base pair reads, we can sometimes see reduced classification at the species level in the data analysis, though this does depend on the species present in the samples. For fungal metagenomics libraries, we recommend the 2 by 151 base pair read length. Both 16S and fungal metagenomics libraries have low base diversity. Since we are amplifying a single target in the libraries, most of the sequencers will be very, very similar and may only differ from each other in just a few bases. Because of this, we spike in higher percentages of the PHYX control library. PHYX in this case not only acts as a sequencing control, it also adds base diversity to the run to improve the sequencing metrics. We also will typically target lower cluster densities for amplicon-based libraries, as this will help to improve the sequencing performance. In the following slides, we will review a 16S run on the MiSeq instrument, run with version 3 sequencing reagents in a 2 by 301 base pair run. Fungal metagenomics libraries will perform similarly to 16S libraries, though as mentioned, we are always using that shorter read length for the fungal libraries. All right, so we will start on our metrics tab in BaseSpace. And first, we will review our passing filter percentage. For this run, it is 88.83%, which is very good. Passing filter percentages can sometimes be lower for amplicon-based libraries compared to tagmentation-based libraries and other shotgun metagenomics library types, though this does depend on the sequencer we're using and how we set up the run. All right, so when we check the aligned percentage, we see that it is 9.36. So 9.36% of the data is going to PHYX. We spike in a higher PHYX percentage when we're sequencing the amplicon libraries because we want to add that diversity that we don't have with these libraries compared to the shotgun metagenomics libraries. And so here, this higher alignment percentage is expected. And then here we can navigate to our charts tab. And we'll check the Q30 percentage first. Expanding the chart here helps us to see a bit better. And here we see our Q30 percentage is 70.1%. Q30 percentages will commonly be lower for amplicon-based libraries compared to shotgun libraries and other highly diverse library types. Also in this case, our read length is very long, with paired 301 base pair reads, so we do expect the quality to be a bit lower. For this type of run, our Q30 percentage of 70.1% is perfect. So we'll go back to our Charts tab, and we'll select our data by cycle chart, and then change our chart type to %base in the drop-down menu. Then we'll go ahead and expand the display by clicking the icon on the top right corner of the plot. We will zoom in a little more. And now we can see our %base plot for the run. So here again, I've labeled the insert and index reads to help us review the %base. Here we see the plot is very, very spiky, and this is expected. Because we are amplifying a single target amplicon, at any given cycle, most of the bases present will be the exact same base. This %base plot is exactly what we expect for 16S libraries, and this is perfectly fine. Fungal metagenomics libraries will also look similar to this. All right, so we've completed our review of the 16S run, and all of our metrics are in line with our expectations, which is great. And so now we can proceed with data analysis. For more information on evaluating the success of sequencing runs, we have a helpful bulletin at the first link here. More information on how much Phi-X is needed when we're sequencing low diversity amplicons on different sequencing platforms can be found at the second link. And we have an application note that discusses sequencing 16S libraries on the iSeq at the third link. More information, including the download for the sequencing analysis viewer software, is here. We have a very helpful recorded webinar for how to use sequencing analysis viewer and review run metrics available on our webinar page. And supporting documentation for all of our sequencers can be found at the third link. This is where we have information on how to set up sequencing runs on all sequencers and additional resources for successful sequencing. So now that our sequencing has completed and we verified that the runs performed well, we can go ahead and proceed with data analysis. In metagenomics data, we want to assess the diversity of each sample and identify the taxa present. There are many analysis methods available, from Illumina, publications, research groups, and even custom analyses. We will discuss some options for analysis today, though the second session of our Metagenomics webinar series will be a comprehensive review of analysis options. For online data analysis, we can head to BaseSpace Sequence Hub. You can store run information, generate FASTQ files, and perform downstream data analysis all in BaseSpace. For analysis workflows, we can select the Apps option, which will be near the top of the page once we sign into our account. There are many different app options for metagenomics analysis. We can select Metagenomics from the drop-down menu near the top right to filter apps relevant to our project. One popular app for analysis is the Dragon Metagenomics pipeline. This analysis is compatible with amplicon-based and shotgun metagenomics sequencing data. The Dragon RNA pathogen detection app can be used for analysis of data from the respiratory virus oligo enrichment panel and other sequencing types. We can also analyze metagenomics data locally, so offline. The local Run Manager software is compatible with iSeq, MySeq, MiniSeq, and NextSeq data. The MySeq Reporter software can be used to analyze MySeq metagenomics data, though most labs have transitioned to local Run Manager or to BaseSpace for data analysis. In addition to these, there are many third-party analysis workflows as well as custom analysis options that may be used. Base-based analysis apps can be found at the first link. Helpful documentation and software downloads for Local Run Manager can be found at the second link, and more information on MySeq Reporter is at the third link. And just as a reminder, this has been a super basic highlight of analysis options. Our upcoming Metagenomics Part 2 webinar for data analysis will cover analysis options and workflows in depth. Let's summarize our discussion today, and I'll include some additional resources to help with your metagenomic sequencing project. Today, we've discussed what metagenomic sequencing is, resources for getting started, library preparation methods, sequencing for metagenomics libraries, and briefly touched on data analysis. We've reviewed resources along the way for more information and in-depth guidance. Public datasets are available in BaseSpace, which give run performance metrics and example analyses. Public data can be found in the Demo Data tab in BaseSpace and can be filtered based on our project interest by searching or selecting categories in the right-hand panel. Additional tech support webinars can be found at the first link. We have upcoming webinars, and you can register for these here. And we have links to recorded webinars on multiple topics, from library preparation to data analysis, which can be viewed at any time. We have helpful support bulletins, which include information on best practices for library preparation, sequencing, and data analysis, along with many other topics. We have publication reviews, which feature applications of Illumina Next Generation Sequencing technology. And you can always reach out to technical support via techsupport@illumina.com or by calling us directly at the number here. This is our training page, which I linked to in the previous slide. Here you can see how we can filter results to view new and upcoming webinars near the bottom left-hand side. Thank you so much for taking the time to join me today. I hope that this has been helpful for you.